Welcome back everyone. In this video, I'll be talking about the processivity of DNA polymerase. It's a very important characteristics of DNA polymerase because processivity means the average number of DNTPs or the nucleotides, that will be added per time of DNA polymerase binds with primer template. So each time DNA polymerase that binds with the primer template, how much is the number, the DNTP that will be added to the template by following the basis of the template strand of the DNA. That is the processivity of DNA polymerase, and it basically associated with this whole processivity or processive nature of the DNA polymerase is basically associated with sliding clamp proteins. With the help of sliding clamp proteins, the processive nature of the DNA polymerase will be increased. And there is another important thing, that almost about 10,000 DNTPs, or you can say the nucleotides also, that can be added per binding event of DNA polymerase to the primer template junction or primer template. So it is highly processive DNA polymerase. If DNA polymerase is non-processive for some other reason, like wrong DNTP if added like that, at least one DNTP definitely is going to be added to the templates. So if DNA polymerase is non-processive in nature, only one DNTP that will be added per binding event. And if the DNA polymerase is highly processive, then it will gonna add about 1,000 DNTPs per binding event. This is the processive nature of the DNA polymerase, which is very much important for efficient DNA replication. Thank you so much.